Hello, everyone. Uh, I'm Tim Hovanec. I'm going to talk about nitrifiers and nitrification today. There's my contact information. I used to be a mud in the boots fish farmer. I was in the Peace Corps, raised in the Philippines, raising milkfish and Penaeid shrimp, and then worked for Aquaculture Systems International out in Southern California that eventually became Kent Seatech, raising hybrid striped bass. And then I went into more research in closed systems. So the traditional view of nitrification is that ammonia, and it is ammonia the bacteria use, is oxidized to nitrite by Nitrosomonas europaea, and then nitrite is oxidized to nitrate by Nitrobacter winogradsky. And that those are nitrifying bacteria. They were first isolated by Sergei Winogradsky over now 120 years ago. And it was kind of that, those are the two, and that's who does this process. It's two-step. Doesn't matter whether it's freshwater, saltwater, low substrate, so low ammonia, low nitrite, or high nitrite. But if you look back in the literature, there were some hints that that really wasn't the case. And then, of course, there's all these bottled mixes, and they never worked. And why is that? So there was some hints out there that this might be the case. And I'm going to show you what the current state of the knowledge of nitrifiers and who they are is, but we're not going to dwell on that because I know this is not a microbiology group. Then it's how do we put this information to use when you're setting up, cycling, and maintaining a biofilter. The short story is in aquatic recirculating aquatic systems, low concentration systems. It is not Nitrosomonas europaea that's doing the ammonia oxidation, and it's not Nitrobacter winogradsky that's doing the nitrite oxidation. I showed first that it's these bacteria called Nitrospira and Nitrosospira. That was 27 years ago, and there's now many groups that have followed on this research. And the reason why everyone thought it was Somonas europaea and Nitrobacter winogradsky is that for a long time, all studies to isolate and study nitrifying bacteria were done on what's culture dependent. You had to take samples and you had to grow the bacteria. And the formulas for those had large amounts of ammonia, over 10, 20, 30 milligrams per liter. And that turned out to be a selective bias, because if you use or give a nitrifying system large amounts of ammonia, which will result in larger amounts of nitrite, these are the organisms that you tend to grow. But I did my Ph.D. at UC Santa Barbara on this topic using molecular methods, molecular probes, sequencing, clone libraries, denaturing, gel electrophoresis, or DNA fingerprinting. These are all culture independent methods. So we could just take a sample and develop these probes to tag or mark the bacteria. And when I took that to a sewage treatment plant, they would light up pretty well, showing that Europaea and some Winogradsky were there. But when I looked at aquarium systems and aquaculture systems, the probes came up blank. And that, long story short, resulted in that there's a wide diversity of nitrifiers, whether they're the ammonia oxidizers or the nitrite oxidizers, and it very much depends, as I'll get into, the environment that you're sampling. There isn't just a ammonia oxidizer or a nitrite oxidizer. And then as studies progressed and we got better at these techniques, what was found, and this is everybody wants to show pictures of their fish. This is actually called fish fluorescent in situ hybridization. using molecular probes. The red is the targeted nitrospira, the nitrite oxidizer. The green is the ammonia oxidizers. And it's hazy because they live in a biofilm. They don't live single-celled. They don't live much in the water. They live on surfaces. They extrude an extra polymer substance, and they live in a biofilm, the importance of which I'll cover in a few minutes. But then in about 2005, it was discovered that there were ammonia oxidizing archaea. Now, the bacteria belong in a different group. Doesn't like when I do over here. Most of them, ammonia oxidizers, nitrite oxidizers, are in the proteobacteria. Archaea are a whole different super kingdom that are as different to bacteria as bacteria are to eukarya. And all the references that I'm going to talk about and give were all done in either aquariums, public aquariums, or aquaculture facilities. So these are all low ammonia, low nitrite fish culture systems. And what this group found was that there were ammonia oxidizing archaea. Then another group found that there is anaerobic ammonia oxidation. It was typical that you had to have oxygen present to have for the nitrobacter, nitrospira, nitrosospira to work, you needed oxygen. But actually, there's anaerobic ammonia oxidation, or ANAMOX. It combines ammonia with nitrite to produce dinitrogen gas. Again, this was from a marine recirculating aquaculture system. The drawbacks to this, one, the environment is anaerobic. The organism is actually a planktomyces, and the intermediate is hydrazine, which is part of rocket fuel and is poisonous to most living organisms. And the bigger problem or issue is that the doubling time for these organisms is 7 to 22 days. So, but they have been found in aquaculture systems. They're just very slow growing, and their importance is probably negligible, but we don't know conclusively. And then about 8 years ago, complete ammonia oxidation. Sorry if this, I'm not touching anything, so I don't know why it's doing this. Nitrification was always considered a two-step process, but if you look at the energy, it could be a one-step process. And two groups at the same time published in Nature that they were able to isolate and show complete nitrification by a single microorganism. And the single microorganism belongs to the Nitrospira sublineage. We're not going to go into any of that, but I have the references if you're interested. But it turns out that there is a clade or a sublineage of Nitrospira that can oxidize ammonia right to nitrate. It's a one-step process. Right now, that only seems to occur in freshwater systems. So the current views are there's aerobic nitrification, and that's ammonia to nitrite, and that is done generally by Nitrosomonas marina, which was originally isolated in salt water, but does live in freshwater, as I'll show. And then you have nitrite to nitrate, and that's done by various members of the Nitrospira group. And then you have ammonia oxidizing archaea, and again, Nitrospira. And then you have the one-step ammonia right to nitrate by this sublineage of Nitrospira. Then you have in anaerobic environments, ammonia, nitrite to nitrate by these to nitrogen, to dinitrogen by planktomyces. But the real question is, okay, if you're a microbiologist, you love all this diversity and finding things, but for an aquaculturist, how do we use this information? What does that tell us? And so in this table, you can see that whether it's a freshwater system, marine system, hydroponics, or aquaponics, these various processes, except coaminox, have been found. it has not been conclusively shown in a full strength seawater system just yet. So they're all there. And that's one thing to realize is that these organisms in microbiology out in the field isn't single species. They're not pure species. They're consortia. And it depends upon many factors of which ones are going to dominate. And that would depend upon the environment you're trying to grow your organisms in. And so which nitrifiers are important? That is determined, number one, by salinity. That is the most important factor determining the consortia of nitrifiers you're going to have is the salinity of your water. Next is temperature. If you're doing aquaculture below 10 degrees, the nitrifying organisms are much different than if you're doing nitrification at 20, 25, 30 degrees Celsius. If you're trying to do nitrification in that, 8 degree to 12 to 15 degree, that's probably the toughest because both groups, they don't like that area. They don't grow well in that temperature, groups being, as I'll show in a minute. Secondly is substrate concentration. This plays an important role in deciding which organisms will dominate. And more importantly, it plays an important role if you're starting a new system to establish the correct organisms, as I'll go through. And then particulate organic concentration. So how much organics you have in your system, which can be important with people that are, if you're doing hydroponic systems or trying to do any of the systems that have a lot of particulate load, that will select for different types of nitrifiers. And then the age of the biofilter. Biofilters, as they mature, are just like most ecological systems. There's R species and K species as the biofilter ages, assuming it doesn't have perturbations or disruptions. I highly recommend this study, and what they looked at was fish culture systems in freshwater, FR, I don't think there's a pointer, or FR, brackish water systems, and then marine systems. And these numbers are operational taxonomic units. The ones in parentheses are unique. So like in brackish water, they were able to identify 3,895, not just nitrifiers, but just different organisms. 2,519 were unique for the brackish. They weren't found in full-strength marine systems. They weren't found in full-strength freshwater systems. Then looking at the nitrifiers, what they found was that salinity had the greatest overall effect on microbial composition and for the nitrifiers. And I also showed that years ago where I could take, and this, my papers are published in Society of Microbiology Applied in Environmental Microbiology. Just Google my last name. But I took functioning biofilter that was freshwater, functioning biofilters that were saltwater, and then changed the salinity. And you'll see they've got the same effect that I got. AOBs were found more than AOAs. Now, when AOAs, ammonia oxidizing archaea versus ammonia oxidizing bacteria, when AOAs were first discovered, the thinking was that maybe they were more dominant. And that's really generally been found not to be the case, except in very special situations. And that Nitrospira is the dominant nitrite oxidizing organism, no matter what the salinity of the system. And you can see the Venn diagram here. And what was super interesting was that the marine nitrifiers exhibited a greater salinity range than the freshwater nitrifiers. And then the Hovanec was found up into brackish water, but not in full-strength seawater. And what this chart is showing from the same research is that this is inhibition. So 0 inhibition to 100% inhibition. In freshwater biofilters, as they isolated those organisms, established them on filters, and then slowly changed the salinity. And as they increased the salinity down here on the x-axis, nitrification was inhibited. Those freshwater organisms cannot be switched to saltwater. Brackish water organisms had a decent range and were inhibited only up to about 65, 70%. But saltwater organisms, starting down here and going fresher and fresher, it wasn't until you got to basically fresh water that there was much demonstration of inhibition. And even in that case, it was only 75%. So the marine nitrifiers have a wide range of functionality over salinity. And that is what we've repeatedly seen in the biofilters that I work with. And the importance of that, I'll demonstrate in a minute. So if you look at, I looked at salinity, now temperature. Depending upon your temperature, when you're down below 10 degrees, this is where the ammonia oxidizing archaea dominate. And that is why mixtures and full disclosure, I do grow nitrifying bacteria, but we're not talking about commercial and how to grow things. But people that want to buy nitrifying mixes, if your system is below 10, you need to talk to your provider about what is the organisms in that mix, because ammonia oxidizing bacteria do not grow, do not do well, do not dominate in systems that are below 10 degrees. And right in here, in that 10 degrees plus or minus is where you get both, and neither works very well. Even at below 10 degrees, the temperature is so low that the bacteria growth is very slow, so it takes a long time to establish the biofilter. Next is effective substrate. Why is it doing that? So in very low substrate concentrations with very clean water, you find the Hovanec. Increased organics seem to inhibit this group. As you go up a little bit, you get the AOAs, and then when the concentration is higher, the AOBs dominate. And how this can work in nature is generally during the first 30, 60, 90 days of establishing a biofilter, your system is dominated by AOBs, but as it ages, some research tends to show, and it's not conclusive, that the AOA seem to play a bigger role. But again, that depends upon the amount of organics that are in the water. And that's why some people will say they have a change, that their biofilter went sour, and it can be due to the switch of these organisms, and that relates to organic levels. And everything I'm saying, this isn't a settled science. There's a lot of research being done. The University of Vienna, there's a whole institute there dedicated just to nitrifying bacteria. And there's a couple of research institutes in Germany too that have a lot of people studying this. So it's an ongoing discovery. But one thing I want to point out is that nitrification, no matter the organisms, takes place in a very thin biofilm. It's only 100 microns. So you've got up here the water layer, and groups using microelectrodes are able to measure oxygen, ammonia, nitrite, nitrate, and as it goes through the biofilm, showing the activity. And the purpose I show that is in all these cases, I keep on talking about organics, you want to make sure your biofilms are kept clean. That's why past studies have shown that rotating biological contactors, fluidized beds, things that keep organics from settling and burying nitrifiers, have those types of biofilters have always worked better. And that's because nitrification takes place in a very small thin film area. So how can we use this? And obviously, a lot of people, it's starting up the biofilter for biosecurity in a hatchery, or it's seasonal when they're running their closed systems, but they're starting up, and how do they want to do that? And they want to do that fast. First off, ammonia concentration. It is better to continuously dose a small amount of ammonia rather than just pour a large concentration in once a day. You want to keep the ammonia nitrogen and nitrite nitrogen below 5 milligrams per liter at all times. I mean, if it gets above it, it doesn't mean the bacteria are going to die. It's very hard to kill these bacteria. But if you culture or start up your biofilter at a high level, 10, 20, 15 milligrams per liter, what you're going to do is get a system dominated by the Nitrosomonas europaea. But then as you add fish and the ammonia levels drop, what we commonly see is that the biofilter crashes and the whole system goes through another startup phase. And that's because, and this is detailed in my paper of mine in 2001, and there's others after that show that there's the Nitrosomonas europaea decline, the filter resets, and then you eventually get the Nitrosomonas marina types. So start out with a low concentration, continuously dosing, and your filter will go much faster. Also, the nitrite oxidizers spend most of the energy generated from converting nitrite to nitrate in keeping the cells intact. So they grow much slower. Their doubling time is about 1.5 times that of the ammonia oxidizers. So typically when you're dosing, the ammonia will disappear quickly, but nitrite will start to build up. You need to resist continually feeding ammonia and getting the nitrite super high, because studies have shown that the nitrite can be poisonous to both groups of bacteria. Nitrite is very poisonous to most organisms. And what that means is you don't have to feed your biofilter every day. We get this even from professionals. Well, I have to feed it. My bacteria are going to starve. They're not going to starve. They can go days without ammonia. So resist the temptation to keep on dosing ammonia that will lead to your nitrite getting sky high, because the whole system will stall, and then you'll just have to do a big water change for the bacteria. Also, get your temperature up as high as you can do. We culture these bacteria at 30 to 35 degrees. And so when you're doing a system above 10, say you're doing 15, it's much faster to cycle if you start out at 30, grow a large number of bacteria, and then slowly reduce the water temperature to your target temperature. That system will cycle a lot faster than if you started at 15 and waited for the bacteria to double. It start high and then reduce the temperature no more than 2 to 3 degrees a day to your target temperature. If you're doing a saltwater system, again, the higher the salinity, the more energy the cells have to put into maintaining cell turgor, the integrity of the cell. But as I showed, the saltwater bacteria do very well in brackish or even freshwater systems. So lower the salinity down to about 22 to 26 parts per thousand, and set your biofilter up at that temperature, or at that salinity. You'll get a bigger population faster. Then you can increase your salinity no more than 5 parts per thousand per day. So just a couple of days to get up to the target temperature or target salinity. Importantly, maintain your pH above 7.5. And doing fresh water, make sure that you maintain your alkalinity. Nitrification produces hydrogen ions. Hydrogen ions will consume your alkalinity and then drop your pH, switching the ammonia to the ammonium NH4 form, and that is not the form the bacteria utilize. And then Also adding a small amount, just a small little bit of orthophosphate and iron. I won't go into the physiology of the cells, but this will help. 0.05, 0.01 for both. Just during cycling, the cycling will go much faster. And resist using ammonia removing chemicals. I can't stress this enough. We make an ammonia removing chemicals. Don't use that. Let the bacteria go to work. They inhibit. They do inhibit the nitrification bacteria. They also, unless you're using a buffered form, drop the pH, which I just said you don't want to do. So resist using chemicals. It's just going to prolong the process. Troubleshooting a biofilter. Things happen. The number one is be measuring nitrite. The nitrite oxidizing bacteria are kind of the canary in the coal mine. If your filter's going sour, if there's something wrong, if the organics are building up and the water, so the oxygen can't get to the biofilter, nitrite will generally show up first. And then check water flow, check your pH and alkalinity. And don't let the organic slime take over and bury your biofilter. Keep your biofilter clean. And then also nitrifying biofilms are food. They're food for many organisms. The thin film works best, and they're tough. I know this might sound like what's he talking about, but biofilter, you can take it out and you can hose it off with tap water. A little bit of chlorine because the nitrifiers are in that biofilm. I'm not saying soak it in chlorine, but to take a hose and just spray it off to clean off the biofilter. The nitrifiers are stuck there really well. And this is actually a big problem in water distribution systems is the nitrifiers or zebrafish systems. The nitrifiers live on biofilms inside the pipe, and they really, that ESP acts as a force field that protects them against chlorine or any medications and things like that. So you can spray them off. One thing that does kill nitrifiers is freezing. You can use expensive cryopreservatives, but if your biofilter freezes, the ice crystals pretty much puncture the cell and kill the bacteria. And then when all else fails, a water change pretty much can solve a lot of things. General conclusions, there are distinct differences between freshwater and marine nitrifying communities. Which nitrifying organisms or consortia dominate depends upon many factors and can also vary over time. Generally, the proteobacteria ammonia oxidizing bacteria dominate. And in lower systems, in freshwater, clean systems, you may get the Hovanec. And how important Animox is still not presently known. There's references. I'm available email, and definitely appreciate your time and take questions.